Texas Lutheran University.
Welcome, ladies and gentlemen, to the combined chemistry and biology seminar. Chemistry first. Today we have presenting Dr. Andrew Hinck. He is a professor at University of Texas Health Science Center at San Antonio. He's a part of the biochem department. A quick history on him. He's from up north. He went and had undergraduate degree from University of Puget Sound. Anybody know where that is? No? Puget Sound, very good. In chemistry, good job. He got his PhD from University of Wisconsin and did 2 postdocs afterwards, where he, I would say, fell in love with NMR in his undergraduate career and went on to go delve further into NMR. He's written numerous articles. I mean, I'd show you and I'd read them off, but he's amazing, pretty much. I got to work with him this summer, and he is awesome. Also, if you have any questions about graduate school or even doing summer programs at the Health Science Center, talk to him afterwards. Just some fun facts, because I love this. This summer, while I worked with him, I rode on a bike 7 miles. So I've got 7 miles per year. This man right here bikes about 8,000 to 9,000 miles a year. So listen up.
All right. I think this is working. Well, thanks a lot for the invitation to come here. It was really great to have Michelle in the lab. She was a really outstanding summer student, and I presume that reflects on her training here at TLU. And I presume there's a lot of other students like Michelle, well, not exactly like Michelle here at TLU, but of academic excellence like Michelle. But anyway, thanks again for inviting me here. My background is in, I was a chemistry student at UPS in Tacoma, college comparable to this. I then went on to graduate school and studied biochemistry. And we're in a lab that focused on NMR spectroscopy of proteins. And then as a postdoc, I also continued in that area with some real experts at the NIH, at BACS, and some other really great NMR spectroscopists. And then I came to San Antonio in 1997 and set up my lab. My lab is primarily focused on the proteins I'm going to tell you about today, which are proteins, signaling proteins of the so-called transfluorine growth factor beta superfamily, or TGF-beta superfamily. I'm going to talk today about some of our work with TGF-beta and one of its co-receptors called beta-glycan. And if I can do this right, this is an overview of the proteins of the TGF-beta superfamily in humans. And there are over 30 such proteins. The ancestors of the protein are these so-called bone morphogenetic proteins, or BMPs for short. TGF-betas are evolutionary latecomers to the superfamily. You find TGF-betas only in vertebrates, whereas you find the BMPs in very simple organisms like worms. There are 3 BMPs in worms. As well, of course, in humans, and there are many of the BMPs in humans. So these proteins, they evolved in simple multicellular eukaryotes, and where they regulated embryonic development. But as organisms have become more complex, they've retained their important roles in embryonic development. But as the protein family has diversified, so too have the functions. So you see, if you come over toward the TGF-betas, you have this group of proteins, the so-called activins. These are important in regulating, these are endocrine hormones that regulate ovulation. You have other proteins that, such as myostatin, that regulate muscle size. You have a protein called MIS that is important in regulating sex determination during development. The TGF-betas here are the proteins which I study primarily. They're important in regulating the adaptive immune system as well as wound healing and so forth. So the point is that these proteins arose in very simple multicellular eukaryotes, but as eukaryotes have diversified and become more complex, the proteins of the superfamily have also duplicated and taken on increasingly more specialized roles. So the focus of my research is to study these proteins and using primarily structural biology, so NMR, but also crystallography and other tools, try to understand what are the molecular adaptations that have occurred both in the signaling proteins, which are shown here, but as well as their corresponding receptors that have enabled them to carry out their functions in a very specific manner. Okay, the reason we want to study these proteins and understand how they function, how they attain their specific functions, is because many of these proteins, I would say almost all the proteins of the family, have some corresponding human disease. So the TGF-betas, for example, are responsible for this disease called Marfan syndrome. This is a disease of vascular fragility. Basically, if you have this, you have a very high likelihood of your aorta rupturing by the time you turn about 20 years old. This is another very dramatic disease caused by a defect in one of the receptors of the superfamily, in particular a receptor for one of the bone morphogenetic proteins of the superfamily. The receptor is mutated such that it has a higher than normal basal level of constitutive signaling activity, and that leads to bone formation in soft tissue. And basically, individuals with that defects in that receptor develop bone at practically all sites in their body, and they basically can no longer live because they can't breathe, they can't move any longer. And that's called this disease here, not this one. It's called fibrodysplasia ossificans progressiva. So these proteins are known to cause many important human diseases. And again, we'd like to understand how they function at the molecular level so that we could develop therapies for treating diseases such as this, or any of the other number of diseases that I have listed there. I'm a biochemist, I said, and the way we approach this is by looking at these proteins and the corresponding receptors at the molecular level using techniques such as NMR or crystallography. So these are structures of some of the proteins of the superfamily. I started working on these proteins when there was just a couple structures of these proteins known. Others have been determined since. They're summarized here. You can see that they're all have these are different representative proteins of the family, and you can see that they all have the same overall fold. They're all homodimers, meaning that they're comprised of two protein chains that are identical, and almost all of them, but not all of them, have their two-component monomers covalently linked together by a single disulfide bond. So you can see that here in TGF-beta1, TGF-beta2, BMP2, and GDF5. So these are covalent homodimers. If you run these proteins on an SDS gel, under non-reducing conditions, they run around 25 kilodaltons. If you rerun them under reducing conditions, they'll run at half that mass. All the proteins of the superfamily also signal through similar receptors. And importantly, all the proteins of the family require two different receptors that are known as a type 1 receptor and a type 2 receptor. And these receptors are structurally homologous to one another. Both the type 1 and type 2 receptors have these relatively small but very highly disulfide bonded extracellular domains of only about 100 amino acids. A single spanning transmembrane domain follows, and then you have a cytoplasmic serine threonine kinase domain. And the type 1 receptor is differentiated by the type 2 in the inclusion of this additional motif that's known as a GS motif. This is basically a regulatory element that, under normal conditions, inhibits the activity of the type 1 receptor kinase. So signaling is initiated when the ligand homodimer, which is shown here schematically in the middle, binds to the ectodomains of the receptors, assembles them into a tetrameric complex with 2 type 2 receptors and 2 type 1 receptors. That brings the kinase domains in close proximity to one another. The type 2 receptor kinase is constitutive. The type 2 receptor kinase, because it's in close proximity to the type 1, phosphorylates serine residues within this regulatory GS motif of the type 1 receptor. When particular serine residues in this motif are phosphorylated, that motif moves out of the active site of the type 1 receptor kinase, allowing it to then phosphorylate downstream substrates. The most important of which are the so-called SMAD proteins. So SMAD proteins are normally cytoplasmic proteins. When TGF-betas, TGF-beta family, superfamily ligands bind to the ectodomain receptors and assemble such a complex, the type 1 receptor becomes activated. That phosphorylates the SMAD proteins. And upon phosphorylation, the SMAD proteins will translocate to the nucleus, where they affect transcription in cooperation with different coactivators and co-repressors. And as I said, all the proteins of the superfamily signal through this same mechanism, although there are subsets of receptors that are specific for specific subsets of ligands. So one of the things that we've been focusing on in my lab is trying to understand, well, why do certain receptors associate with certain signaling ligands and vice versa? And I'm going to tell you in the first part of my talk about that, what we learned about that. So yeah, so along those lines here, these are the different type 1 and type 2 receptors of the superfamily in humans. So you have 7 type 1 receptors and 5 type 2 receptors. These are the rough pairings of the signaling proteins with the corresponding receptors. So you see, for example, TGF-betas bind this pair of type 1 and type 2 receptors that are called TBR1 and TBR2. Whereas the BMPs and GDS generally pair with one of these type 1 receptors and one of these type 2 receptors in different combinations. And one of the important things is that the type 1 receptors that these signaling proteins couple to and activate, the SMADs that these type 1 receptors couple to and activate is different than the SMADs that these type 1 receptors couple to and activate. And this is actually kind of important because, as I mentioned, TGF-betas are among the most recently evolved members of the superfamily, whereas the BMPs are among the most are the ancestral ligands of the superfamily. So basically, what you see if you just look at kind of crude biochemical pairings of receptors with ligands, you see that the ancestors of the superfamily couple to type 1 receptors that then activate a different subclass of SMADs than the more recently evolved members of the superfamily. So TGF-betas will bind type 1 receptors that activate SMADs 2, 3, whereas BMPs will bind to type 1 receptors that then activate SMADs 1, 5, 8. So you see a distinct pairing of signaling proteins with receptors, and then in turn you see a distinct subclass of cytoplasmic effectors that are activated. So we want to understand the molecular basis. Why is it that TGF-betas only bind the TGF-beta type 1 and type 2 receptor and not the BMP type 1 and type 2 receptors, and vice versa, using structural biology, so NMR and crystallography. So to do that, I had a very talented graduate student, PhD student in my lab at the Health Science Center, Jorge Zuniga, and then later also a very talented graduate student, Tao Huang, come into my lab and work on this project. And another individual who worked on it was a postdoctoral fellow with expertise in crystallography, Jay Grappi. So all three of them worked together on this project in different aspects. And we started by focusing on TGF-beta and the TGF-beta receptors, and in particular, the higher affinity of the two TGF-beta receptors, which is the TGF-beta type 2 receptor, or TBR2 for short. So this is the NMR spectrum of the TGF-beta type 2 receptor extracellular domain. The thing about these receptors, as I mentioned, is that they have many disulfide bonds. This receptor has 6 disulfide bonds. To get the protein to study it by NMR, we want to produce it in E. coli so we can make it in an isotopically labeled form. So that means we have to somehow produce it in E. coli. When you produce this protein in E. coli, it forms inclusion bodies. It's insoluble. It doesn't fold. So you have to renature it. So we worked out a renaturing procedure for this receptor. And then we recorded the NMR spectrum. And what we saw was a very nicely dispersed pattern. We're here in this spectrum. It's a proton N15 shift correlation spectrum. So we're looking at, in this case, this is an N15 labeled protein. So we're looking at the backbone, mainly we're looking at the backbone amide resonances of this protein. So we know the size of that protein. You basically expect then to see a corresponding number of spots in this NMR spectrum. And you also expect them to be well dispersed. The dispersion of the signals in a protein NMR spectrum is indicative of folding. So if the protein's unfolded at random coil, all the NMR signals will be condensed on top of one another. When the protein folds up, you get unique magnetic environments for each of the amides, and the spectrum becomes dispersed. So this was a very, this NMR spectrum itself basically told us that the protein is almost certainly natively folded, because we, A, saw the expected number of spots. We didn't see like 2 or 3 or 4 times the number of spots, which would be indicative of heterogeneity. And we also saw that the spots were very nicely dispersed. So even without performing any kind of functional assay, the NMR spectrum of this protein was telling us that it was natively folded. Not guaranteeing that it was natively folded, but indicating that highly likely that it was. So then we started looking at functional assays. Here we're looking at binding, the ability of this receptor to bind TGF-betas, purified TGF-betas. So here we're just using a very simple native gel. So this is a non-denaturing gel where we mix increasing molar ratio of the TGF-beta type 2 receptor extracellular domain with a fixed molar ratio of the TGF-beta homodimer. And what you see is as you add increasing molar amounts of the R2 relative to the TGF-beta, you see the appearance of this complex. This is the R2 alone. The TGF-beta alone does not run in the gel, and this is the complex. And you see that complex increases in intensity up to the point where you've added 2 equivalents of R2 relative to TGF-beta homodimer. And then when you go beyond that, you then start seeing no further increase in the complex and an excess of the R2. So this is really very nice data because it's showing that the R2 that we refolded can bind the TGF-beta homodimer with high affinity, high enough affinity to remain associated during the time course of the electrophoresis, which is on the order of an hour. And furthermore, that it binds the TGF-beta with 2-to-1 stoichiometry, which is what's expected based on the fact that you have a monomeric receptor and a dimeric ligand that it's binding to. So to be more quantitative in terms of the affinity of this purified receptor for the TGF-beta homodimer, we performed a surface plasmon resonance experiment. In this experiment, we couple TGF-beta to the surface of a gold sensor surface, and then we inject over that using a microfluidic system, increasing concentrations of the purified R2 extracellular domain. We see a very nice, robust, concentration-dependent response. We can fit the maximum amplitude at equilibrium as a function of concentration to derive the KD, and we derive a KD of about 200 nanomolar for this purified receptor binding to the TGF-beta homodimer. So that's further indication that indeed this is a very natively folded protein, because you would never have a natively folded receptor that was binding its corresponding ligand with an affinity of 200 nanomolar. Okay, so that was good. So then we went on to work on the TGF-beta type 1 receptor extracellular domain. This is the other one. It turns out that this domain was previously known not to bind TGF-beta with very high affinity on its own. It was seen to interact in cell-based studies only very weakly with TGF-beta alone, but it would bind with high affinity to the TGF-beta type 2 receptor complex. So we made this TGF-beta type 1 receptor extracellular domain. This also has a lot of disulfides, 5 in this case. We also, when we produced it in bacteria, it was also insoluble. We worked out a renaturation procedure for this, recorded the NMR spectrum, and again saw a very nicely dispersed pattern with the expected number of spots indicating that this protein was natively folded. We then performed native gels, similar to what I explained to you before. It's a little bit complicated, but here, basically what we did is we first tried combining the type 1 receptor with the type 2. That's, for example, in this lane, you see this is the type 2 receptor. This is the type 1 receptor by its alone, by itself. When you mix them two together, you just see the same two bands. So the type 2 receptors, when mixed together, don't associate with appreciable affinity. If you mix the type 1 receptor with TGF-beta, which is in this lane here, so the TGF-beta alone doesn't enter the gel. The type 1 receptor is here. When you mix them together, you just see the type 1 receptor. So again, the type 1 receptor does not appreciably bind with high affinity to the TGF-beta homodimer. Now, if you go and look at the binding of the type 1 receptor to TGF-beta, but in the presence of the high affinity receptor TBR2, the situation changes. So here in this lane, lane 6, you have the complex between the high affinity receptor TBR2 and the TGF-beta homodimer. This is the same thing that I showed you on the previous slide. So this is the high affinity receptor bound to the TGF-beta homodimer. When we add the type 1 receptor to that, which runs here, this band upshifts and gets a lot darker, meaning there's more protein in the complex. And that's presumably the ternary complex. So what's interesting about this gel is it shows that the type 1 receptor is binding with high affinity to the TGF-beta type 2 receptor TGF-beta complex, but the type 1 receptor doesn't bind well to either TGF-beta alone or the type 2 receptor alone. It binds well, but it binds with high affinity to the R2 TGF-beta complex, but not to either component alone. And that was very satisfying because here in this case, we're just performing this experiment with totally purified proteins that we refolded, and that behavior is recapitulating what had been reported earlier for these receptors based on cell-based studies. So our purified proteins are recapitulating what had been seen in cells on the cell surface. So that's really good because then if we were to determine structure of this complex per se, for example, that should tell us how TGF-betas bind their receptors and how they cooperatively assemble into a ternary signaling complex. I won't go through it, but we also were able to validate these interactions of the type 1 receptor to the TBR2 TGF-beta complex using SPR. OK, so I'm kind of skipping a lot here, but after a while, we were able to ultimately crystallize that ternary complex, so TGF-beta homodimer bound to the type 2 receptor and the type 1 receptor, both in 2-to-1 stoichiometry relative to the TGF-beta homodimer, and then determine the structure. So that structure that we determined experimentally is shown here. You have the TGF-beta homodimer in the middle, blue and red. The type 2 receptor ectodomain is in green, and the type 1 receptor ectodomain is in yellow. And I'll tell you a little bit more about that structure, but one thing I want to say is that other groups at other research institutions were carrying out similar studies with BMPs, and those groups were able to determine this structure shown here. This is of a structure of a BMP homodimer bound to its corresponding type 2 receptors and its corresponding type 1 receptor. And importantly, even though I'm not showing you here, these groups in addition to determining the structure, also performed similar binding studies, what I explained to you for TGF-beta, but with BMPs and the BMP receptors. And in that case, they found that the binding of one receptor to a BMP had no influence on the binding of the other receptor type. So in contrast to what had been seen with the TGF-beta. So in the BMP case, the binding of one receptor doesn't seem to substantially affect the binding of the other receptor type, in contrast to what's seen with TGF-beta. And once we got these structures, it kind of became obvious why the TGF-betas differed in the way they bind their receptors compared to BMPs. And that is that if you look at the receptor complexes, you see that the type 1 and type 2 receptor are in direct contact in the TGF-beta receptor complex, but not in the BMP receptor complex. In the BMP receptor complex, the type 2 and type 1 receptors bind at independent sites, and the receptors don't touch one another. There's no contact between them. Whereas in the TGF-beta receptor complex, in the TGF-beta receptor complex, you have this intimate contact between the type 1 and type 2 receptor. So we posited that it's this direct receptor-receptor contact that's responsible for the pronounced stepwise assembly of the TGF-beta receptor complex. That is, the type 2 receptor binds TGF-beta with high affinity on its own, and then that basically completes the binding site for the type 1 receptor, with the type 1 receptor binding not only to TGF-beta, but also to the type 2 receptor. And I'm not going to show the data on that, but we were able to validate that by mutating residues in the interface between the type 1 and type 2 receptor, as well as making some deletions of this part of the type 2 receptor, which corresponds to the N-terminal tail. In that case, we found that the type 2 receptor was perfectly capable of binding TGF-beta, but it was very impaired in terms of its ability to bind and recruit the type 1 receptor into the complex. So why do I bother to tell you all this? I bother to tell you all this because this is actually kind of not the expected result, which I think is the coolest thing about doing science, is when you do something and you think it's going to turn out one way, and then it actually turns out some other way. And I think it's then when you actually learn something new. And in this case, I think we learned something new that I think it was a surprise not only to me, but to really everybody in the field. So the surprise is that you have these two very different architectures for these receptor signaling complexes. But the thing is that all the proteins that are involved in forming these complexes are structurally homologous to one another. So I already told you that TGF-betas have the same fold as BMPs. You can see that again here. They're both homodimers. They both have this fold where each monomer, which takes the shape of a hand, packs like that. They both look like that. Furthermore, if you overlay the folds of the type 2 receptors for TGF-beta and BMP, or the type 1 receptors for TGF-beta or BMP, you see that those folds are also the same. So here you can see, in fact, this type 1 receptor for TGF-beta is the same protein, basically. There's differences in some loops, but it's basically the same protein. Same for the type 2 receptor for TGF-beta and BMP. So that's kind of unexpected then, that you have proteins that have the same folds, yet when they bind and form these signaling complexes, they're really very different. And they're really different. It's not like it's a subtle change. It's not like a slight movement. They're entirely different. So for example, here, you can see for the BMP, it uses this so-called concave, this beta sheet surface, to bind on what's called the knuckle of the ligand. Whereas in the TGF-beta receptor complex, the type 2 receptor actually uses this edge beta strand right here, and it binds into what's called the fingertips of the ligand. So you have a different interface for the type 2 receptor for TGF-beta and BMP, both on the ligand side, but as well as on the receptor side. So it's not like a subtle shift. It's an entirely different interfaces on both sides, which, as I said, was not really anticipated by anybody. So that's what we learned, is that TGF-betas, the evolutionary latecomers to the family, bind and assemble the receptors in a way that's very different from the ancestors of the family, the BMPs. So that's summarized here. So here we have showing the two different modes of type 2 receptor binding, either BMP or TGF-beta, relative to a common ligand. And you can see that they're really different. It's not subtle at all. The difference between the type 1 receptor is a little bit more subtle. And, the type 1 receptors turn out to use the same beta sheet surface to interact with the ligand, although the way the type 1 receptor is positioned on the ligand is quite a bit different. So there's about a 10 angstrom shift between the centroid of where the BMP type 1 receptor binds compared to the TGF-beta type 1 receptor. So in this case, the type 1 receptor is using the same surface, but the region of the ligand that's contacted is actually quite different. So you have basically, among receptors that have the same fold, whether you're talking about TGF-beta type 1 or type 2 or BMP type 1 or type 2, there's only one receptor fold. And yet, among that one receptor fold binding to ligands, you see 4 different modes of binding. So that's actually a really surprising result. And so we think this is important because, as I said, if you look at the TGF-beta superfamily, you kind of have this, you have 2 clades. You have the ancestral clade that bind type 1 receptors that activate SMADS158. And you have the more recently evolved clade that includes the TGF-betas, but as well some other proteins that bind type 1 receptors that activate SMADS2 and 3. So we think that what happened over the course of evolution is the ligands and receptors are duplicated, but instead of keeping a common interface between the ligand and the receptor, at some point, a new interface evolved. And we think that that's important for expanding the range of specificity. Because if you're duplicating ligands and receptors, but always keeping the same interface, the only variable that you have is to vary the residues in the interface. So you can get some specificity by doing that clearly. If you put some hydrophobic residues here, then you want to match it with hydrophobic residues on the receptor or charged residues or whatever. You can play that game and you get so far, but that's only going to give you a certain range of specificity. If, on the other hand, you create an entirely new interface, then the residues on both the ligand side and the receptor side can now differently evolve for the TGF-betas versus the BMPs. And you can get very high specificity and effectively complete segregation of signaling for these proteins relative to these proteins. So we think that's what's happened, and we think that's an interesting finding from the work that came out of my lab as well as some other labs over a period of about 10 years. I'm not sure how I'm doing in terms of time. So let's see. Okay, so I want to tell you one more story, and then I'll finish up. So this next story has to do with the fact that so I've told you how you have these 2 different clades, and that the ancestral proteins and the more recently evolved proteins of the superfamily activate distinct subclasses of SMADs. It turns out then that explains specificity to some extent, but really only kind of to a really limited extent, actually. It's only a mechanism for discriminating the activity of these proteins from these proteins. But how are the activities of all these proteins discriminated from one another, or all of these proteins discriminated from one another? Because all of these proteins over here bind type 1 receptors that activate SMADS158. All of these proteins over here bind receptors that activate SMADS2-3. How do you discriminate all of these from one another, or all of these from one another? So that's what we've been working on more recently. And the thinking there is that, that it's not the unique signaling among for example, if you focus on this clade here, it's not the unique signaling per se that's elicited by these family members that determines their unique biological activities. It's instead what cells and tissues those ligands are targeted to. Because that's another way of getting specificity. If you target certain ligands to certain cell types or tissue types, you're going to get a unique activity compared to other proteins, even though the intrinsic signaling is the same. So that's the thinking there, is that there are mechanisms in place that guide the different proteins of the superfamily to different cells and tissues, and they respond in unique ways, and that's how you get the unique activities of proteins among one clade relative to one another. So What are the proteins that guide the proteins? What are the proteins that guide these signaling proteins to specific cells and tissues? Well, there's a lot of them known. Those are shown in blue here. So these are the so-called accessory binding proteins. And there are almost as many accessory binding proteins of the superfamily known as there are signaling proteins, as you can see here. So these proteins are quite abundant. They're quite diverse. So all these proteins have different structures. And so the idea is that with these different structures, these proteins recognize unique features of each of the different signaling proteins. The signaling proteins are all similar, but they're not identical. And the idea is that you have all these diverse accessory binding proteins, and those recognize specific features of the different signaling proteins, and in turn that targets them to specific cells and tissues. That's important for their activity. So what I want to tell you now about is this accessory binding protein known as beta-glycan. This is an accessory binding receptor for the TGF-betas. It's been known for some while. And this is kind of shows why studying this protein might be important. So if this is an experiment where, not done by me, reported by others, where you take radiolabeled TGF-beta, so iodinated TGF-beta, and then you just basically throw that at cells and then add a cross-linker and look to see what proteins cross-link to the radiolabeled TGF-beta. So if you do that, you see the type 1 and type 2 receptors, which I already told you about, and which we now know the structures of, and the receptor complex and so forth. But you also see this really big blob here, and it's pretty dark. This is the type, this accessory binding protein for TGF-beta called beta-glycan. It's also called the TGF-beta type 3 receptor. It's a proteoglycan, so you have a core protein as well as a glycosylated form with glycosaminoglycan chains that are highly predominant. So there's a lot of beta-glycan. If you do this on most cells, you see a lot of beta-glycan compared to the signaling receptors. So what is this protein doing? It turns out it's a very high affinity TGF-beta binding protein. It binds all three TGF-betas with high affinity. And it has the interesting activity that it helps one of the TGF-beta isoforms, TGF-beta 2 in particular, bind and assembles its receptors into a signaling complex. So in humans and other vertebrates, there are three TGF-betas, TGF-beta 1, 2, and 3. It turns out that TGF-beta 1 and 3 bind the TGF-beta type 2 receptor with high affinity, whereas TGF-beta 2 isoform does not. So TGF-beta 2 turns out to have low intrinsic affinity for the, quote, high affinity TGF-beta signaling receptor, TBR2. And accordingly, cells are not very responsive to TGF-beta 2 relative to TGF-beta 1 and 3, unless the co-receptor is present. So TGF-beta 2 binds with an affinity that's about 1,000-fold lower than beta 1 and beta 3 to the type 2 receptor. And cells are about 1,000-fold less responsive. So it takes about 1,000-fold higher concentrations of TGF-beta 2 to elicit the same response as TGF-beta 1 and 3 if beta-glycan is absent. But as shown here, if you take those cells and you repeat the experiment with beta-glycan now expressed, either by transfecting it or just using a natural cell line that naturally expresses beta-glycan, you see that those cells are now equally responsive to TGF-beta 2 compared to TGF-beta 1 and 3. But the beta-glycan itself is not a signaling receptor. It's just a TGF-beta, it's a membrane-bound TGF-beta binding protein. So how does it do that? How does this TGF-beta binding protein that's on cells but can't signal make cells respond and increase their responsiveness to TGF-beta 2 about 1,000-fold? What's the mechanism? So we were interested in trying to understand that. We still don't know the answer to that. We have some specific ideas. I'm not going to, in the interest of time, I'm not going to go through all the experimental data on this. But let me just say that there's, based on the previous cell-based data, there's a proposed model. The proposed model is that the beta-glycan, which is membrane anchored, binds TGF-beta 2 and forms a complex on the cell membrane. And then that complex in turn somehow recruits the type 2 receptor, which has, the type 2 receptor has low intrinsic affinity for TGF-beta 2. But somehow the association of beta-glycan with TGF-beta 2 promotes the binding of the type 2 receptor. How is not known, but that's what we're trying to figure out. So you form this so-called presentation complex here, and then it was proposed based on, this is all based on experimental data, that the beta-glycan is displaced as the type 1 receptor is then recruited into the complex to form the signaling complex. Again, this is all based on cell-based experimental data, but not with purified proteins. So we want to go back now with purified proteins and see if we can understand the mechanism in molecular detail. So that was the job of another PhD student in my lab, Maria Villarreal, who was from South Texas. So Maria made various domains of beta-glycan and then studied their binding properties using SPR. So this is the anatomy of beta-glycan. This is actually a pretty big protein. The ectodomain of beta-glycan is about 75 kilodaltons. It has a single spanning transmembrane domain and then a very short non-structured cytoplasmic C-terminal tail. The ectodomain of beta-glycan can be broken down into 3 structured domains. This so-called N-terminal E domain or endoglin domain, and then membrane proximal uromodulin domain. And that itself is comprised of 2 tandem immunoglobulin-like domains, it turns out. So you have 3 independently structured domains. The GAG chains are attached to this first of the immunoglobulin-like domains as part of the U domain. It turns out that this domain by itself can bind TGF-beta, as well can the membrane proximal domain. This domain is a non-binding domain. Okay, so that's the anatomy of the protein. How is it that you have a protein like this with one TGF-beta binding domain here, another down here, can promote assembly of the receptor signaling complex with TGF-beta 2? So Maria made full-length beta-glycan ectodomain, as well as all the component binding domains, purified them, and then studied their binding properties using SPR. And I'm not going to go through all the data because there's quite a bit, but I'll try to summarize. So this is one experiment that Maria did. And one of the first things that Maria was trying to do was try to determine the stoichiometry. How many beta-glycans are bound per TGF-beta homodimer? So what Maria did was she made a SPR chip with TGF-beta mobilized on it, and then injected over that mobilized TGF-beta the high-affinity TGF-beta receptor, TBR2. And you can see it binds very nicely. She could measure the binding affinity. And then she repeated the same experiment with the very same chip, with the very same TGF-beta on it, but using the beta-glycan extracellular domain. So in this case, the full beta-glycan extracellular domain. And then she looked at the response. Again, this receptor was binding very nicely. In fact, it binds with higher affinity than the type 2 receptor, about 5 nanomolar, compared to about 130 for the type 2 receptor. But one of the things that she did was she looked at the amplitude of the response. So in SPR, the amplitude of the response is proportional to how much is bound, the fraction bound, but also the mass of the thing that's bound. So if you have something that binds its 10 kD, and then you have something that's 100% occupancy, and then something that's 100 kD that binds 100% occupancy, you'll get a 10-fold higher response for the 100 kD partner compared to the 10 kD partner, just because of the larger mass. So what Maria did was take the mass out of the equation by plotting what's known as the mass normalized SPR response, rather than just the raw SPR response. So she's taking into account the fact that the mass of this receptor is a lot bigger than the mass of the R2 receptor. So doing that, what she then sees is she then compares the maximal amplitude of the SPR response normalized for mass with the full-length beta-glycan extracellular domain. You see that the response is roughly one half that of attained with the type 2 receptor alone. And we know from our previous structures that the type 2 receptor binds TGF-beta homodimers with 2-to-1 stoichiometry. So what this SPR data is telling us is that the full-length beta-glycan extracellular domain differs from the type 2 receptor and that binds TGF-beta homodimers with 1-to-1 stoichiometry rather than 2-to-1 stoichiometry, as for the isolated R2 extracellular domain. So I'll just tell you about one more experiment because I think this is the most interesting one. So then Maria did another experiment. In this case, again, she's looking at type 2 receptor binding. But she looked at type 2 receptor binding for immobilized TGF-beta alone or immobilized TGF-beta alone in the presence of a near saturating concentration of a full-length beta-glycan extracellular domain. So this is just binding the type 2 receptor to TGF-beta alone. Again, you get a very nice concentration-dependent response. You can fit this and get the maximal amplitude, which is shown in black here. If you repeat the experiment, the only difference now being that you have a saturating concentration of full-length beta-glycan extracellular domain in the buffer, and then look at binding of R2, you see now actually that the maximal response at saturation is, again, approximately 1/2 that of R2 alone. So what this tells us is that in the presence of beta-glycan, you can only bind 1 R2 instead of 2, as is normally the case with a bare homodimer. So if beta-glycan is bound, then TGF-beta changes such that it can now only bind 1 R2 compared to 2 for the wild-type homodimer without any beta-glycan bound. So that's interesting. So that suggests then that beta-glycan's binding with 1-to-1 stoichiometry relative to homodimer, but it binds in such a way that it leaves a spot for 1 R2 to bind. Okay? It doesn't block out both R2 binding sites, it only blocks out one. And that's kind of cool because mechanistically, the job of beta-glycan, as I told you, is to somehow potentiate binding of this low affinity TGF-beta to the type 2 receptor. So I'm not going to show you all the rest of the binding studies, but there were a lot of additional binding studies done. I'm going to skip through those and just go to this because it summarizes the data that I didn't show you as well as the data that I did show you. So basically, what was proposed on the data that I did show you is something like this. Okay, so you have the TGF-beta homodimer. We think what happens is, owing to the very high affinity of beta-glycan for TGF-beta and the high abundance of beta-glycan on the membrane, that basically captures this TGF-beta that has a normally low intrinsic affinity for the type 2 receptor and concentrates it on the cell membrane. Okay, because beta-glycan binds very well to the TGF-beta 2. And there's a lot of it, as I showed you. So we think that that's one of the primary jobs of beta-glycan is to capture this TGF-beta 2, this ligand that has low intrinsic affinity for the TGF-beta type 2 receptor, concentrated on the membrane. Once this complex is formed, the beta-glycan TGF-beta complex, remember I told you that when beta-glycan binds, it binds in such a way that it leaves a spot for one type 2 receptor to bind. So what we think happens is that the beta-glycan captures the TGF-beta, concentrates it on the membrane, and that promotes the binding of the type 2 receptor because now basically you've overcome one of the entropic degrees of freedom for binding. So now for the type 2 receptor bind, you only have to overcome 2 degrees of freedom to form the complex rather than 3 degrees of freedom if you just had TGF-beta 2 free in solution. So basically you're lowering the entropic barrier to complex formation. The formation of protein complexes, the major barrier is the decrease in the entropy. Okay, so you're overcoming the entropy, the unfavorable entropy, at least to some extent. So we think it's basically an entropic mechanism that promotes the formation of that ternary complex. And then in data that I didn't show you, this domain, the E domain, the N-terminal domain, binding domain, has to be displaced for the type 1 receptor to bind. So we think that's kind of a critical step because when this domain is displaced, and then the type 1 receptor binds, remember I told you from the structure of the TGF-beta receptor complex that when the type 1 receptor binds, it doesn't bind to just TGF-beta alone. It binds to a combined surface formed from the TGF-beta type 2 receptor and TGF-beta. So the idea here then is that as this N-terminal binding domain is displaced by the type 1 receptor, the type 1 receptor comes in, and then that's going to essentially stabilize the bound R2 because all these interactions are going to reinforce one another. So just as binding of R2 to TGF-beta potentiates the binding of R1, binding of R1 will also potentiate the binding of R2. So all these interactions will then reinforce one another. And then at this point, you just have this one domain of beta-glycan bound. So we think, because now no longer do you have this high residence time due to multivalent binding, the beta-glycan we think is then displaced, and then you form the signaling complex. So we basically think that it's interesting that the beta-glycan, it's probably, well, the thinking is, we don't really have hard data on this, this is something that we need to do and want to do, is basically functioning like a catalyst. So basically we think that increasing the rate at which the TGF-beta2 homodimer binds and forms the ternary signaling complex with the type 1 and type 2 receptor. We think that the rate of formation of this complex would be a lot slower in the absence of beta-glycan, because the problem is that kinetically, it would be slow to form and accumulate any significant population of TGF-beta bound to the type 2 receptor, but in the absence of beta-glycan. So we think that we don't have all the pieces there, and we also don't have all the structures to validate where these domains are binding. But again, we think it's the beta-glycan kinetically potentiates. It translate binds the TGF-beta, but that is then displaced as the signaling receptor complex is formed. OK, so I'm going to skip this part too. We're working to get the structures using a combination of NMR and crystallography. This is the NMR spectrum of both of the binding domains of beta-glycan. These are a lot bigger proteins than the signaling receptors that I showed you before. But with the instruments that we have at the Health Science Center, we think we can get the assignments and then the structures of these proteins. And this is work being done by another newer PhD student in the lab, Kate Kim, who came to my lab from UCSD. We're also trying some crystallography as well. This is some crystals that are formed from a complex between TGF-beta, the type 2 receptor, and the N-terminal binding domain of beta-glycan. And if somehow I got invited back here, I hope I would, but if I were to get invited back here, say in 2 or 3 years, I would really hope that by then we would have these structures and we could show you in molecular detail how these domains are binding. And then this is some more NMR work trying to map the interfaces and build up the complexes as an alternative approach to the crystallography. I'm not going to go into that. And I think I've already summarized how I think beta-glycan works, so I'm not going to belabor that further either. So I just want to say one last thing, is that I'm at a health science center, and the goal of doing this basic research, as I said from the outset, is really to try to understand how these proteins bind and assemble their complexes, with a goal of hopefully, ultimately, this information one day leading to some development of some therapeutic that helps treat human disease. So one of the things that we've been working on is trying to develop TGF-beta, using the structural information that we've gained to try to develop effective TGF-beta inhibitors for cancer treatment. So I won't go too much into detail, but one of the things that TGF-beta does is it's a very potent immunosuppressive protein. So it turns out that because of that, many cancer cells will produce very, very high levels of TGF-beta. And the data is that what's happening is that TGF-beta is effectively deactivating all the different type of immune cells that would normally recognize the cancer cells as foreign and destroy the cancer cells. So that's for cancer cells to proliferate and for the tumor to expand. It basically requires that the immune system be deactivated. So cancer cells are being formed all the time. They're normally recognized as foreign and destroyed by cells of the immune system. But for tumors that form, somehow they get past a critical window, and then they start producing proteins like TGF-beta that then suppress the immune system, that then allow the cancer cells in the tumor to expand unchecked by the cells of the immune system. So TGF-beta is, for this reason, but for some other reasons I'm not going to go into, has arisen as a really important target for cancer therapy, in particular for cancer immunotherapy. So basically the idea is to develop inhibitors that would bind and neutralize the activity of TGF-beta, the excessive TGF-beta being produced by cancer cells or a tumor. And then that would then essentially allow the immune cells to be reactivated and then basically come in and destroy the tumor. So the therapy would not be killing the cancer cells directly. It would just be neutralizing the TGF-beta so the immune cells can be activated, and then the immune cells will do the dirty work and do it cleanly of destroying the tumor. So there is some very good preclinical data showing that you can actually do that. So you can take animals, you form very large tumors, treat them with very effective TGF-beta inhibitors that activates an immune response, and then you can destroy or regress very large tumors in a very short period of time. You can then re-inject those animals with the same cancer cells that you initially used to form the tumor, same number, same cells, and those cancer cells cannot form a tumor at all. And that's because now the immune system recognizes those cancer cells as foreign and destroys them even before they have a chance to form a tumor. So this could be a very cool and very powerful new form of cancer treatment, where you go in and you're not trying to kill the cancer cells, because if you try to kill cancer cells, you always kill normal cells, and that's bad. It's just trying to reactivate the immune system so the immune system can get rid of the tumor for you. So I won't go into it, but there's been a lot of effort because of the things that I'm telling you in pharma to develop effective TGF-beta inhibitors for cancer treatment, including kinase inhibitors that target the active site of the TGF-beta type 1 receptor kinase, mainly by Lilly. These have been clinical trial for over 12 years now and are still in phase 1 trial. So I'm actually kind of skeptical that they're ever going to be approved for clinical use. And this is, I think, mainly due to off-target activity. There's also an antibody that was developed by another pharma called Genzyme in Cambridge, Massachusetts. This antibody is very specific for TGF-beta, but it turns out it's not very effective. And I think the reason it's not known why it's not effective. It's been used in phase 1 and phase 2 human clinical trials. It's very safe, but it's not very effective. So one idea that I have, the company, when something like this doesn't work, the company doesn't publish, well, we think it's not working very well because of this or this. They just don't publish that. So one idea that I have is that the antibody does not have high enough affinity to compete against the endogenous receptor complex for binding TGF-beta. So the antibody that Genzyme developed binds TGF-betas with affinity of about 5 nanomolar, whereas the TGF-beta receptor complex, which is a heterotetramer, and due to multivalent binding, actually will bind TGF-betas with picomolar or subpicomolar affinities. So it's not clear to me that at the doses of therapeutic antibody that you can put into an animal that you can achieve high enough concentrations to basically outcompete the very high affinity, very avid cell surface receptor complex for TGF-beta. So that's one idea, is that the antibody just can't compete against the cell surface receptor complex. So we've been trying to work to develop alternative approaches. And one idea is actually a very simple idea, but one idea that I hope might actually work is to develop these so-called soluble receptor fusions. So this is where we take the ectodomains of either the signaling receptors, R1 and R2, or beta-glycan, and then fuse them together with flexible linkers, but in a way where those receptor ectodomains can still bind TGF-beta in the way that they naturally do. So the idea is to try to recapitulate the multivalent binding of TGF-beta by the cell surface receptor complex, but with recombinant proteins that are soluble, and we can inject into animals, and that hopefully will bind the TGF-betas with very high affinity, like the cell surface receptor complex, but then essentially encapsulate the TGF-beta and sequester it and prevent it from signaling and neutralize it that way. So that's the concept. Oh. Uh-oh. Help. OK, so my time is up. So I'm going to just show you this one thing. This is just some SPR data right here with one of the so-called soluble receptor fusions that we've developed. It's a trimeric fusion. So it has 3 binding domains connected together by flexible linkers. So it's 2 R2s and then the N-terminal binding domain of beta-glycan. We call it RER for short. This is the SPR trace of binding of RER to TGF-beta. You can see that it binds relatively rapidly, but that this is the dissociation phase of the SPR experiment. So we're no longer injecting the R2 at this point here. So you can see that the receptor binds, this soluble receptor fusion binds, but then it basically doesn't let go, or it lets go very slowly. So it has a very slow off rate. So that's really what you want for a good inhibitor. You want it to bind, but then not to let go. And it's, from these SPR measurements, we can see that these receptors, because they have the off rates, the on rates are reasonably fast, but the off rates are relatively slow, that they're binding with very high affinity. Exactly how high affinity they're binding is actually hard to tell because it's so high affinity. But this is a cell-based experiment now where we're looking at neutralization of TGF-beta in a cell-based assay. So we have increasing concentrations of the inhibitor with a fixed concentration of TGF-beta. You can see that the EC50 for this inhibitor is in the sub-picomolar range. This is the log of the nanomolar. So it's between -3 and -4 of log nanomolar. So that means it's a sub-picomolar inhibitor for TGF-beta. So I'm not allowed to tell you that because we actually now have a collaboration with this company. I can tell you that. We have a collaboration with a company that's trying to develop immune-based therapy for TGF-beta for cancer treatment. And we're collaborating with them. They're interested in our inhibitors as an alternative to the antibody or kinase inhibitors. And those results are ongoing. And hopefully the company will, the hope is the company believes that that molecule that we've developed has potential to be developed into a therapeutic for human use, which takes a lot of money. So the company looks at these different possible therapeutics very carefully and assesses the strengths and weaknesses because they're going to have to put a lot of money in to develop it into a human therapeutic. So they're taking a hard look at ours right now. We don't know yet whether they'll invest in it, but I'm hopeful that they will. We're also trying to develop some novel small molecule inhibitors that are highly specific. This is a project that Michelle was working on in the lab this past summer. That project is under development. Hopefully we'll have some of those some specific small molecule inhibitors as well. These would be not targeting the kinase. These would be blocking assembly of the complex in the first place. Okay, so I was asked by Dr. Toledo to just say a few words about my career path because I think sometimes people come in here, professors like me, and say, okay, we're running this lab and doing all this stuff, a lot of publications. How does this happen? Could I do that? And so I just want to tell you basically how I got to where I am. I don't think I did anything magic. So I do want to point out a few things that I think were important for me in terms of my career development. So as I mentioned, I was an undergraduate student at the University of Puget Sound in Tacoma, Washington. This is a picture of the campus. It's a very nice campus there. You can see it's very woodsy, and this is the Puget Sound in the background there. I was a chemistry student, and the department was relatively small. I think there were 9 professors in the chemistry department. One of them happened to have a pretty strong background in NMR, inorganic chemist, and he had an advanced NMR course. I took that, and and they had a pulsed NMR instrument there. So I started, kind of getting some fairly detailed information or training in terms of the theory of NMR, but then also how to operate the instrument, kind of hands-on. And I think that was actually pretty important. And I also challenged myself. I took challenging courses because I knew that when you go into your career, you don't have time to go back and learn linear algebra or some physics or these other things. The time to learn that is really when you're in college. So I really took that opportunity to take some challenging courses. So I took challenging courses in not only chemistry, but also physics and math. And I also, this turns out because of the proximity to Seattle and a company in Seattle called Microsoft, that this university had a pretty strong computer science program. So a lot of my colleagues ended up getting jobs for Microsoft. And one of them actually is the leader now for the Excel program that maybe some of you have used. But a lot of my students were there, so I took advantage of that because it's kind of obvious, even when I went to college in 1983, that computers were going to be important for the rest of my career. So I wanted to take advantage of that. But I think the most important thing is participating in research as an undergraduate. So I applied for and attended a summer research program at Oak Ridge National Lab in Tennessee. And then I also then continued that research project for my senior research project back at the university. And I think that research experience is really important because when I was a PhD student at the University of Wisconsin in Madison, you can see it's a bigger place, I went into a bio-NMR lab. And there were a lot of students. There were a lot of postdocs. It was a big lab. We had about 20 people in the lab. My advisor was very well established. And because of my research experience, when I went to the lab, I was pretty comfortable working in the lab. And even though the kinds of things that were going on in that lab were different from what I had done in my undergraduate research, I just felt comfortable being in the lab, and I could kind of get started right away working on the projects that were kind of given to me in the lab. And I was able to, within being in the lab 1 year, publish a paper in JACS, and then subsequently 3 other first author papers in biochemistry, a number of second author papers. I would say another key was choosing the right supervising professor. I think there's an advantage to choosing established investigators who have a reputation and also have a record for publishing publishing the results consistently. And my advisor had those characteristics. I think he was a good advisor. Some PIs are very slow. They don't publish that much. They're not as established in their research communities. I think that's a detriment to someone who's trying to get their PhD and establish themselves. So anyway, I then ended up because of the work that I had done as a student, basically, I ended up in another NMR lab, very good NMR lab at the NIH in Bethesda, Maryland. NIH has a really outstanding bio-NMR community there. Maybe some of you guys have heard of a person named Ad Bax. Ad Bax is a really outstanding NMR spectroscopist. He's developed a lot of the methods that are widely used for studying proteins these days. I got to work closely with Ad as well as several of his colleagues. It was a really great environment. I also, being at the NIH, that's how I got to work on TGF-beta. TGF-beta is a medically very important protein. It was discovered at the NIH. I got to work with Anita Roberts, whose group was the one that discovered TGF-beta. So that was important too. So I got to not only additional NMR experience, but I also got working on a medically relevant problem, which then I think then helped me get the job here in San Antonio because there are groups here already in San Antonio interested in TGF-beta. So that's basically it. I'm sorry to go over a little bit. I've already acknowledged the people that did the work. It was, again, a combination of PhD students and postdocs. So thank you very much for your attention, and I'm happy to take any questions.
So your gels, your polyacrylamide gels are native gels. They're not SPS gels, is that right?
The gels that I showed, yeah, those are native gels. So you just leave out the SDS.
Right.
Yeah.
That's why your TGF-beta didn't enter on.
Yeah, it has the wrong charge. Yeah. Those native gels are nice. You know, it's not very quantitative, but if you just want kind of quick yes or no answers, are you forming a complex or not, it's just really easy to do. If you go in to do an SPR experiment, it's a lot more complicated. The chip costs $400, and then you have to do a bunch of operations to run the instrument. It's just a lot more complicated to get your result. If you just want to know yes or no, you can just quickly do a native gel. Other questions?
Talk a little bit about the information you're bound to get from crystal structure and NMR, because all those interactions that you're looking to get.
Like a crystal structure versus an NMR structure?
Yeah, like the complementary nature of those two things.
Yeah. So it's a little bit of a complicated question. But we have seen instances where the structure that we get from crystallography is not representative of what we have in solution. There's actually another project that we just recently published in biochemistry. I didn't have time to talk about that today. But one of the TGF-betas we were studying, TGF-beta 3, turns out it was originally crystallized. So these TGF-betas we talk about the monomers as taking up the shape of a hand. And there's a helix here, and what's the palm. So the helix, the palm of one hand packs into the heel of the other. So with TGF-beta 3, as well as all the other proteins of the superfamily, they had all of them were done by crystallography. And they all appeared in this kind of canonical way where the heel of, or the palm of one hand packed into the heel of the other in a complementary way. So they would call this a closed dimer. But, and TGF-beta3 crystallized like that as well. But then we subsequently looked at TGF-beta3 by NMR, and we found that actually, if you look at the signals from the palm region, actually we could tell by NMR that it's actually not structurally ordered, that those were actually random coil. And then my group subsequently determined another different crystal structure of TGF-beta3. And in that crystal structure, for the first time, and the TGF-beta was found not to be in this canonical closed form, but instead of this open form like this, and where the monomers had rotated away from one another by over 100 degrees, and there was no electron density for the palm, the helix. And that's corresponded exactly to what we saw, the region where we saw no electron density, the palm, that was corresponding exactly to the region that we saw by NMR that was structurally disordered in solution. So apparently, TGF-beta3, it's not that this original crystal structure of TGF-beta3 in the closed form was wrong, it's that TGF-beta3 is apparently undergoing an equilibrium between the closed and open form, and that the lattice contacts in the original crystal structure just happened to trap and stabilize the closed form. But actually, if you look at it in solution, really, the reality is that equilibrium is really probably in favor of the open form. So that was interesting. So there can be things like that where lattice packing can distort the structure. But generally speaking, if you don't have something like that going on, you determine the NMR structure versus the crystal structure, they'll look very similar. But I would say if you had a choice, if they're like equally hard to do, I would go for the NMR structure. Maybe I'm biased because of my background, but I really do think the NMR structure would be better because you get a lot of information about the dynamics that's just totally absent from the crystallography. I mean, you can infer certain things about the dynamics from the crystallography, but it's pretty crude to what you can do by NMR. NMR can really tell you in intimate detail about the amplitudes and timescales of molecular motion at all sites in the protein. So it's really a more information-rich technique. But it turns out it's a lot harder. You know, it's not actually equally hard to get a crystal structure or an NMR structure. It's a lot harder to get the NMR structure. So that's not always done. But maybe it'll become easier in the future, and then everything will be switched to doing NMR. Other questions?
No more questions. Let's thank Dr. Hinck one more time.
For more information, please visit tlu.edu.